Hello, grade 11 students. In this video, we're going to talk about our new activity about mitosis and meiosis. So, here biology is the only science where multiplication and division, they have the same meaning. They have the same concept, increasing the number of cells. You have to know that mitosis is the process by which the somatic cells, the diploid cells, the 2M cells, they reproduce. They increase in number and they produce new daughter cells, conserving the genetic information from one generation to another. This is the process of a mitosis. So you have to know that mitosis occurs only for somatic cells, for the diploid cells, 2N cells, where they reproduce and they produce new daughter cells. They are genetically identical. They have the same genetic information. Why would the body cells need to reproduce? We have two goals for reproduction or for the mitosis process. First goal is to increase the number of cells in the individual, like the case in the transformation from the zygote to an embryo. So we need to increase the number of cells. Also to renew the body tissues and replace the old ones or the dead cells by new one. Mitosis occurs continuously inside the body. It may be divided into interphase that has 3 checkpoints. We have the G1, it's called gap 1 phase. We have S phase, it's called the synthesis phase. And we have G2, it's called the gap 2 phase. And we have the process of mitosis, it's called the MET. Prophase, Metaphase, Anaphase, and Telophase. So first, we have G1, as I said, gap 1 of the interphase. We have S phase, where the DNA synthesis occurs, and we have the duplication of DNA. G2, it's only the preparation of the cell to enter the mitosis process. It is only preparation. Then we enter into mitosis. We have prophase, metaphase, anaphase, and telophase, and cytokinesis. So we have first, as I said, gap 1, first gap phase. The cell is doing this everyday job. The chromosomes, they are made only of one strand. Then we pass through the S phase, synthesis phase, where DNA is replicated, duplication of DNA, and the amount of DNA increased to double. And during the G2 phase, the gap 2, with the second gap phase, it's only the preparation for mitosis. The chromosome, they have 2 strands of DNA. And then we enter into mitosis, we have 2 daughter cells. These daughter cells, they are identical to the mother cell and have the same genetic information. So let's move now to the interphase. In the interphase, we have the chromatin form. The chromosomes, they are in the chromatin form. So this is the interphase. Prophase, the nuclear membrane disintegrates. So the nuclear membrane around the cell disintegrates. The myotatic spindle fibers, they are formed. The centrosomes change into asters and move to the opposite pole of the cell. The chromatin condense, and we have chromosomes, each made up of 2 sister chromatids. So these are the phases of prophase. The events, we have the nuclear membrane disintegrates. The myotatic spindle fibers, they are formed. The centrosome, they change into asters and they move to the opposite poles of the cell. Chromatin condense, so that the chromosome, they are made up of 2 sister chromatids. Metaphase, as you know, metaphase, the chromosome, they line at the middle of the cell, forming what's known by equatorial plate. So the chromosomes, they line up at the middle, at the equator of the cell, forming what's known by equatorial plate. Mitosis also, we have anaphase. Anaphase, the spindle fibers, they pull the sister chromatids of each chromosome apart. Each chromatid moves to the opposite pole of the cell. This is what we call it, the polar ascension. By the late anaphase, we have 2 identical groups of chromosomes, each made of 1 chromatid at the opposite pole of the cell. Then we have telophase and cytokinesis. Cytokinesis, we have the chromosome form 2 identical groups, as shown here. They form 2 identical groups at the opposite pole of the cell. The chromosomes will decondense, will move back into the chromatin form. The nuclear membrane will reform around each set of chromosomes. The spindle fibers disintegrate, and the asters return to the centrosome state. The membrane constricts, forming what's known by the cleavage furrow. This is the cleavage furrow here, where the cells are constricting and dividing. This is called the cleavage furrow, and is only exclusive for the telophase. And here we have two notes. You have to know that the process when the cytoplasmic membrane starts to constrict and division, this constriction is called the cleavage furrow. This is called the cytokinesis. This is referred to cytokinesis, where the cleaving the cell into two identical daughter cells is referred to as cytokinesis. Okay. Second note, at any stage during mitosis, the cell has two end chromosomes. The two daughter cells obtained, they are identical to each other, and they are identical to the mother cell, and they have the same genetic information. Okay, these are the figures showing the mitosis. Maybe the course I'm going to give you these figures. You have to know that this phase for example, for interphase, you have to know this for metaphase, for anaphase, and this for telophase and cytokinesis. Okay, this is the cell at the end of interphase. Now we're going to move for the meiosis. So interphase is similar to any interphase, but you have to know that interphase in meiosis, it occurs only once. So let's start first with meiosis. Meiosis, we have to talk about meiosis, that it's a process by which the mother cells of gametes will produce gametes. So the mother of cells of gametes is diploid, it's 2N, produces the gametes haploid 1N. Before meiosis begins, the mother cells of gamete will go through an interphase in preparation for meiosis. It includes two successive division, meiosis 1, meiosis 2, reduction and division, and the equation and division. Why it's called reduction? Because the amount of DNA is reduced into half. Why it's called equation? Because at the end of meiosis, the four daughter cells, they are identical and they have the same genetic information. This is a prophase 1. If you see here, we have pairing of the homologous chromosome. This is the difference between meiosis 1 and the mitosis. You have pairing of the homologous chromosome. This is called by synapse. You have formation of tetrads. Why tetrads? We have 4 tetrads. Number 4. So this is the sister chromatids, and we have pairing site of the crossing over. So nuclear membrane disintegrates, disappear. The spindle fibers, they start to form. The centrosomes, they change into asters. Chromatin condensed so that individual chromosomes are now individually distinguished, each made of 2 sister chromatids. And the homologous chromosomes, they pair up. They pair up meaning that they synapse, forming what's known by tetrads. What does this lead? This lead to the increase in the chance of the crossing over between the chromatids. Moving to the metaphase 1. Metaphase 1, we have metaphase in the mitosis chromosome line in the middle of the cell forming equatorial plate. But here, we have something different. No chromosomes, we have tetrads. So the tetrads, they line at the middle of the cell. Whatever we have at the middle of the cell, eventually it's going to be metaphase. Which metaphase? If you have tetrads, it's metaphase 1. Okay? So tetrads, they line up at the middle of the cell, forming the equatorial plate. Then at anaphase, we have polar ascension. Here, not the sister chromatids are pulled by the spindle fibers. The spindle fibers will pull the homologous chromosomes of each tetrad apart. The homologous chromosomes are pulled apart. Each chromosome moves to the opposite pole of the cell. So polar ascension by late anaphase, the result will be 2 groups of chromosomes, each made up of 2 chromatids at the opposite pole of the cell. This is the anaphase 1. Moving to the telophase 1, including the cytokinesis, here we have something special. This is the cleavage furrow. So, during telophase 1, cell membrane constrict, forming the cleavage furrow. So the 2 daughter cells obtained carries 1 copy of each chromosome, made up of 2 chromosomes each. So the obtained daughter cells, they are haploid. They are 1N. Number of chromosomes is reduced. So as 2N becomes 1N. So that's why it's called reductional division. So the meiosis 1 is called reductional division. The daughter cells are different from each other, and they are different from the mother cell. The chromosomes form 2 groups at the opposite poles of the cell. The spindle fiber disintegrates, and the aster return to the centrosome state. The nuclear membranes reform. Note that there is no interphase between meiosis 1 and meiosis 2. You have interphase at the beginning, then meiosis 2, then completely we'll start with meiosis 2, not the interphase again. So prophase 2, you have 2 daughter cells. So prophase 2, the mitotic spindle fibers start to form. Pay attention here. During meiosis 2, it's very similar and very identical in its phases to that of the mitosis. So meiosis 2 and mitosis, they are approximately the same. The mitotic spindle fibers start to form, the centrosome, they change into asters, and the nuclear membrane disintegrates. During metaphase 2, here not the tetrads, here the chromosomes, they line at the middle of the cell forming the equatorial plate during metaphase 2. And anaphase 2, the sister chromatids are pulled apart, each chromatid moves to the opposite pole of the cell, forming what's known by polar extension. By late anaphase, the result will be two groups of chromosomes made up of one chromatid at the opposite poles of the cell. So, Muhammad, if you have any question, we can ask. You want to ask it now, or you can leave it for end?
Yes.
Yes.
هنرجع نعيده للميوسيس ميتوسيس.
Yes, meiosis and mitosis is required for the next sessions. And for example, you have to know mitosis and meiosis for the next sessions. That's why we're repeating them for this year. It's for grade 12 to have the information, to repeat the information, and to fix it. Because in the coming sessions, you have to use meiosis and mitosis again. Okay?
ماشي اوكي.
Okay. So by late anaphase, the result will be two groups of chromosomes, each made up of one chromatid at the opposite poles of the cell. The telophase 2 and cytokinesis. We have the telophase and cytokinesis. The chromosome form two groups at the opposite poles of the cell. These chromosomes will decondense back into the chromatin form. The nuclear membranes start to form around each set of chromosomes, and the spindle fibers they disintegrate, and the acid return to the centrosome state. This is a very important table regarding mitosis and meiosis. You have to know that mitosis occurs in somatic, while meiosis occurs in the gonads. The aim of mitosis is for the growth and the renewal and replacement of the cells, whereas the meiosis is production of haploid 1N gametes, one division only producing two daughter cells, two successive division producing four gametes. So whenever you have four daughter cells or four cells, meaning that it is the meiosis. So now mother cell is 2N and daughter cell is 2N, the same number of chromosomes per cell. Number of chromosomes is reduced into half during meiosis, and going from the mother cell 2N to the produced gamete or 1N occurs to somatic cells in all body organs, occurs to mother cell of gametes in the gonads, made up of one division preceded by interphase. Here it's made up of two divisions preceded by interphase. No pairing of homologous chromosomes. There is no synapse, no formation of tetrads during mitosis, while we have pairing of homologous chromosomes in meiosis. Daughter cells obtain 2N, one chromatid per chromosome, while after meiosis 1, daughter cells 1N, two chromatid, and after meiosis 2, 1N, one chromatid per chromosome. And note that meiosis 2 is similar in its event to mitosis. Why did we repeat this? This is found in the curriculum of grade 12, so we have to repeat this because the next session, inshallah, we're going to start with spermatogenesis and oogenesis. How did the sperm, how is the formation of the sperm occur, and how does the formation of the oocyte or the egg cell occur? So you have to know mitosis and meiosis, and you have to fix the information about mitosis and meiosis before we start with these activities about formation of the sperm. It's called spermatogenesis, and formation of the egg cell oocyte is called oogenesis. So before we start into these, you have to know the mitosis and meiosis. You have to review this, even though they are required in the grade 12 curriculum. So you have to know, even though we have them in grade 11. So it's an additional information to fix them again, because next time I want to speak about the mitosis and meiosis. I'm going to start immediately with spermatogenesis and oogenesis. Next week is going to be about spermatogenesis, the formation of the sperm, and so on. So you have to be careful about this, because these notes they are required during the whole chapter, first chapter, and chapter 15 and 16 for grade 12. So you have to know mitosis and meiosis. That's why I wanted to repeat this in order to fix the information. Okay?